Today we will talk about polymerase chain reaction. Polymerase chain reaction, commonly known as PCR. This important technique has had a major impact on molecular biology by allowing scientists to amplify and study specific DNA sequences. Throughout this presentation, I will explain how PCR works, its application, and its significance in different fields. So basically, what is polymerase chain reaction? Polymerase chain reaction is a widely used laboratory technique in molecular biology that allows the amplification of a specific DNA segments. It can generate millions to billions of copies of a particular DNA sequence from a small sample, enabling researchers to analyze and study DNA with greater detail and accuracy. Here we will talk about types of PCR. Basically, PCR have so many types, but here we will talk about main three types. The first one is the conventional PCR. Basic PCR used to amplify DNA, and the amplified product of conventional PCR is usually analyzed through gel electrophoresis after the process is completed. The second one is the reverse transcription PCR or RT-PCR. This PCR starts with RNA instead of DNA, and RNA is converted into complementary DNA like cDNA by reverse transcriptase before PCR amplification. It's commonly used to study gene expression or detect RNA viruses. The third one is the real-time PCR, which is called qPCR or quantitative PCR, also known as quantitative PCR at measure DNA amplification in real time. That's why it's called real time, using fluorescence dye or probes. This method not only amplifies DNA but also quantify it by tracking fluorescence in each cycle. Here we will talk about the uses of PCR. So the first one is the medical diagnostic. PCR is used to detect infectious disease like COVID-19, HIV, hepatitis, or tuberculosis by amplifying some specific viral or bacterial DNA or RNA sequences. It is also used for genetic testing and identify inherited disorder or some mutation. The second one is the forensic science. PCR play a critical role in the DNA fingerprinting and forensic investigation. Even tiny samples of DNA from hair or blood or skin cells can be amplified to identify individuals and establish biological relationship. For example, paternity test or some crime scene test like that. The third one is the molecular biology research. So the PCR is widely used for cloning genes, sequencing DNA, studying gene expression, and then creating genetically modified organism. It helps scientists study mutation, gene function, and the behavior of specific DNA sequences. The fourth one is the agriculture. How PCR can be used in the agriculture. So PCR helps detect genetically modified organisms and crops and identify pathogen or disease in plants. And it also support breeding program by analyzing specific genetic traits. In agriculture, also when plants getting some stress and for example some salinity stress or heat stress, cold stress, and then we are checking the gene expression through the PCR. Also, when we make the GMO plants like the genetically modified organism or genome edited plants, so we have to confirm that is it GMO or not. So for that purpose, we are also doing the PCR. The fifth one is the environmental science, that how the PCR can be used in the environmental science. So used to monitor microbial population in various ecosystems like soil, water, or air by amplifying and identifying specific DNA sequences from environmental samples. PCR is applied in detecting pollutants and necessary biodiversity. Here we will talk about qPCR. As I discussed before, that we have so many types of PCR like conventional PCR, RT-PCR, and then qPCR. but this video is focused on qPCR. So what is qPCR? Quantitative PCR or real-time PCR? And why it's called real-time PCR? Because it can show the amplification in real time, so that's why it's called real-time PCR. uh, Quantitative PCR is a powerful technique used to quantify the amount of DNA or RNA via reverse transcription in a sample, and it involves the detection of a fluorescence signal during the amplification process, allowing for real-time tracking of DNA quantity. So due to the fluorescence signal, we can check the amplification in real time. What are the key features of the qPCR? Why the qPCR is different from the other PCR? So the key features are, the first one is the fluorescence. Fluorescence dye like cyber green or some fluorescence probe like Techman probe bind to the amplified DNA and emit light, which can be measured during each cycle. So this is the key feature. And then the quantification, the cycle threshold CTValue, which I will explain later that what is the CTValue, is used to determine the amount of initial DNA. A lower CTValue indicate more starting material. This CTValue, this is very important. So I will talk about this later. And the third one is the application, quantification of gene expression, viral load detection, and genetic mutation, and some microbial identification. So the here we will talk about the reagents required. That what are the reagents which are required for the qPCR? Or setting up a quantitative PCR in the lab is a straightforward process. So let's walk through the steps involved. So the essential reagents you will need include DNA or cDNA. Uh, for some PCR we need DNA, for some RNA, but in qPCR we need the cDNA. The the second one is the forward and reverse primer of the specific genes. We need this one. And the third one is the DNTPs. We need the DNTPs for the amplification process. The fourth one is the cyber green and polymerase master mix. These are the reagents required for qPCR. So now workflow of PCR, that how the PCR is working. So and PCR involve 20 to 40 cycles of repeated temperature changes known as thermal cycles, and each cycle typically include three steps. The first one is the denaturation, annealing, and then extension. And these cycles repeats for 30 to 45 times. Like 30 to 45 cycle is depend on the experiment and depend on the PCR, but 30 to 45 cycle it can be repeated. Here we will talk about how PCR works, like how this denaturation and then annealing and then extension can be done. So here we will talk about this. So first this is the denaturation. When the temperature, the reaction mixture which contain the target DNA, primer and DNA polymerase and some nucleotide, as we discussed before, that what are the reagents needed. So and for example in the tube we put the DNA or cDNA, primer, and then DNA polymerase and then some nucleotide, and then it's heated to around 94 to 98 degrees C. So the high temperature causes the double stranded DNA to separate into two single strands. After this is the annealing step. The temperature is lowered to around 50 to 70 degrees C, allowing start synthetic DNA sequences called primers to anneal to the complementary sequence of the target DNA. These primers define the region of a DNA to be amplified. Here also another thing is I will mention about uh, that a probe also anneal to the some part of the DNA. Here there are two parts of the probe. One is the quencher and one is the fluorophore. So I will talk about what is quencher and what is fluorophore. So a quencher is a molecule used in fluorescence based assay like qPCR to to suppress the fluorescence of a nearby dye fluorophore. For example, this R, you can see this is the fluorophore. So it actually at work is to emit the light, but the quencher work is to suppress this fluorophore from emitting the light until it is not detached from this DNA segment. In qPCR, a fluorescence probe such as Techman or Preet-based probe contain a fluorescent dye. At one end is a quencher and at the other is the fluorophore. So the quencher suppress the dye fluorescence through non-radio energy transfer, preventing background signals. So this during PCR, when the probe is cleaved, like when the during the PCR process, when the probe is cleaved by DNA polymerase, the dye and the quencher separate and then allowing the dye to fluorescence that time. So before that the dye will not fluorescence, but when it cleave the DNA polymer by DNA polymerase, so then the dye will fluoresce. So this increase in fluorescence is proportional to the amount of DNA amplified and then enabling real-time quantification. This is the extension step, and the last step is this extension. So the temperature is raised to about 72 degrees C, which is optimal for DNA polymerase enzyme. The polymerase enzyme add nucleotides to the 3' end of the primer, synthesizing a new strand of DNA and complementary to the template strand. During extension, the probe cleaves, like here is the extension. You can see. So here during the extension, the probe cleaves, separating the quencher and the fluorophore. So as I discussed before, that during the extension, the probe cleaves and the separate the fluorophore and the quencher. So the fluorescent signal will be now. I mean the fluorophore will emit the light, so that can be detected by the machine. So finally, the enzyme Tech polymerase synthesized two new strands of DNA identical to the origin of DNA strand. And in this process, the process of denaturation, annealing, and extension constitute one cycle, and under optimal condition, at each cycle of the number of DNA target sequence is double. And here in this these steps are repeated, as I mentioned before, that these steps are repeated for 30 to 45 cycles, so loading to more than 1 billion exact copy of the original DNA segment. So after PCR, after completing PCR, the PCR product is detected by measuring the fluorescence produced during amplification. As I discussed before, that during the cleavage process, the fluorophore then emit the light. So the CT cycle or the threshold cycle threshold or CQ quantification cycle, as I mentioned before, that this is the very important thing. So we will discuss it more deeply, that what is the CT value and what is the CR, what is the CQ value. So the CTU, this is also called cycle threshold or CQ, which is the quantification cycle. So value represent number of cycle required to detect a fluorescence signal above a set threshold. So here, mm, uh, as we can see, this value is important because it indicates the relative abundance of the target DNA. For example, if the red arrow represent gene A and the green arrow represent gene B, so the lower CT value for gene A means it reaches the detection threshold faster than gene B, indicating a higher abundance. So in contrast, the green arrow with a higher CT value takes more cycle to cross the threshold, suggesting it started with less DNA. So the CT, the red curve is have the high abundance, while the green curve have the low abundance. So the higher the CQ or CT value, the lower the DNA quantity. Here now let's take a look at the specific program used in the qPCR machine and what they mean. This is what a typical qPCR program look like. I mean, this is the temperature setting and these things like the denaturation, annealing and extension and how it works. So we will talk about this. So at 95 degrees C, the DNA strand separates, which is the denaturation steps, as I mentioned before. So this happen in any PCR reaction. Next is the annealing step, where primer binds to their specific target's region, and then after this is the extension. This is the key process occur in step 4. During this phase, cybergreen binds to the DNA and the machine detect the fluorescence during this extension step. And after the reaction is complete, a melt curve analysis is performed, and we will soon understand why the melt curve is important. So in the next part, I will talk about this. So let's imagine we have a qPCR and we have completed 30 cycles or 40 cycles, and by the 40 cycle the reaction reach a plateau, meaning we have a certain amount of DNA. After that, temperature is gradually and increase in 1 degree increment. This is called melt curve. So as the temperature rises, the PCR product begin to melt, causing the DNA strand to separate, and at a specific temperature the DNA strand fully separate, and cyber green also, which also separate, which bound to double strand DNA, so also separate, detach. And its unbound state, cyber green, will not fluorescence anymore, so resulting in a sudden drop in the fluorescence in the graph. This temperature, which the fluorescence drop, is known as the melting temperature. So this is the graphical representation of the data output from the qPCR machine. Like when the qPCR is finished, so we can get the graphical representation of this kind of data. So there are the three key types of curve to consider. The first one is the uh, amplification curve or amplification plot. This show whether amplification occurred, and so for example, in this example, the left graph has a cycle threshold CT value around like 22, and while the right graph has a CT around 30, with a cycle represented on x-axis, like we can see from 2 to 40 cycles. So from this we can determine which gene is more or less abundant, as we mentioned before in the previous slides. The next one is the melt curve. So the melt curve, which revealed differences in melting temperature. Here we can see different melting temperature. So on the right, the gene melt at around 85 degrees C, while on the left the melting temperature is closer to 76 or 78 degrees C, like that. So these melt curve can also be visualized as derivative, so showing the genes, showing the changes and fluorescence over time, which create melt peaks. So this melt curve, the importance of these melt curve peaks lie in confirming that there is only one amplicon present. So this is crucial for ensuring that your amplification is a specific and not random, making it an essential step at the end of qPCR. So here we will talk about the qPCR data analysis. Like after the qPCR finish, so there will be some data. So this is the qPCR data analysis. So let's talk about like this data analysis which we get after the qPCR, um, which can be done with various methods. There are various methods using for the data analysis, and one of the popular is the relative quantification known as delta delta CT or method or Levick method. So like for example, imagine we are looking at the CT value of for example any gene, like this is the gene. So we are looking for the gene expression of this gene. So the first one is the control, then the second control, and then the first treatment, and then the second treatment. So uh, uh, like the gene value, we can see the control 1 and control 1 are actually biologically same, but actually these are the technical replicates. And then control 2 and control 2 are also technical replicates, and similarly treatment 1 are technical replicates. So the gene value are presented here. Then we have the actin value. Actin is actually the housekeeping gene. So for every plant and every organism, we have a reference gene, a housekeeping gene. So we are using that gene and as a reference gene. So so after this, the gene also have their own value, and the actin also have their own value. So uh, so we have to find, we have to measure the delta CT value for each sample. So for when we are checking the delta CT value, so the delta CT value we can we can find by uh, the actin minus from the actin value, we will minus from the gene value. We will subtract from the gene value the actin value, so we will get the delta CT value. The next we will calculate the average delta CT value. For example, if there are two technical replicates, as I mentioned before, the control 1 and control 2, so they are not biologically different, but they are just technical replicates. So then we will take the average of these two control. And after this, like so we measure the average of the delta CT of each control. And after that, there is a calibrator. What is calibrator? Calibrator is actually the average of all control CT values. Like for example, we have two control for actually we have two control. So we will take the average of these two control, and so that will be the called the calibrator. And after this, we have to find the delta delta CT value. So which is different from the delta CT of each sample and the calibrator value. Here we will how we will find the delta delta CT value. So the delta delta CT value can be found by we will subtract the calibrator value from the delta CT value of each sample. So then we will find the delta delta CT value, and then from that we will finally we will find the fold change or 2k minus delta delta CT. So we will, this is also called fold change, which we are using in making the graphs and these things. So the formula for delta fold change is 2k power minus delta delta CT, which is part of the Levick method. So this fold change can then be plotted on a graph. So here we can see the graph. For example, the control fold change, the control fold change like it generally be close to 1, around 1, and the treated one is like between the zero point 0.0 and 0.5. So this indicate the gene changes in the gene expression. So this method is essential for researcher and some other uh, plant breeders or agriculturists and forensic and other all the things, to find the gene expression. So this is the end of this lecture. I hope everything will be clear, but still if you have some question you can ask me in the comment section. I will try to answer those questions, and in the next video, in the next lecture, we will practically do the qPCR, that how to make the plate, how to make the cDNA, and how to design the plate for the qPCR, and then how to run the qPCR. We will all these things all we will do practically in the lab. So in the next videos I think it will be then more clear. This data is actually just as an example data, but later on I will do the qPCR practically, and then I will show you the real data which we get from the qPCR results. So then we will also use data, and we will find the fold change, and then we will make the graphs. So I hope you will learn from these things, and if there is any question you can ask me in the comment section. Thank you for listening.